My name is Mitchell Kronenberg, and I'm the President and Chief Scientific Officer. I want to welcome you to another edition of Coronavirus Updates live from the laboratory. I'm going to give you some welcoming and opening remarks, and then we're going to hear from Dr. Erica Ollmann Saphire. And then, if the technology cooperates with us, we're going to go behind the scenes with our Executive Vice President and COO and Steve Wilson, and we're going to look at we're going to be looking at some of the new instruments that we have, and then we'll have a question and answer that will be moderated by me and others. So it's a very special presentation. Also, I should mention that Ruben Diaz-Avalos will be demonstrating some of the new instruments that we have that are going to help us make progress in fighting this infection. Okay, so here we are. I want to just put our effort in some context. We are the La Jolla Institute for Immunology, and Dr. Saphire is our newest group leader or faculty member. She's been with us for 1 year, and we're a relatively young organization. We were founded in 1988, and we have 21 laboratories, about 480 employees. We have postdoctoral fellows, MDs or PhDs from all over the world working on a variety of important problems. And our funding is shown on the lower right here. It comes from a variety of sources, including federal grants, corporate money, university affiliates, and of course, philanthropy. Our mission is to engage in a world-class biomedical research program with a focus on the immune system, and to share that program so that we can make outstanding contributions to the benefit of human health. So why focus on immunology? For many years, I felt we were crying in the wilderness, but now everybody seems to know immunology is really important, and people are slinging around terms like herd immunity, which is good to know that that immunology is appreciated by the public. The immune system is related to so many different diseases. Not only does it protect us from infection, but when it misbehaves, if you will, it can cause chronic inflammation, autoimmune diseases, allergies and asthmas. And of course, gearing up the immune system to fight cancer is extremely important and is really the new generation of cancer therapies. So we've organized our research around disease areas into infectious disease and vaccine research, autoimmunity and inflammation, and cancer immunotherapy. And all of our work really relates to one or more of these areas. And as I mentioned, we have 21 group leaders, and some people's work fits into more than one group. So you see more than 21 people here. So we have a group of 8 investigators who in a relatively short time have begun to focus their attention on this important pandemic infection caused by SARS-CoV-2. And their names are shown here, and some of them have spoken in previous versions of this series of updates, coronavirus updates. And there will be more. So the next one, in fact, will be given by Vijay after Dr. Saphire's presentation. So, why do research if vaccines are already in clinical trial? And our feeling is that SARS-CoV-2 is going to be with us for a while. This is just a plot of the cases in San Diego County. You can see it's not really going down because we don't have all the recent cases yet. And we know that not only are there more cases, but at least in the United States, there are more hospitalizations. So it's not just more testing. We hope that the first vaccines are successful in trial, but we suspect that this is going to be a long haul, and we're going to need years of research to fully control and understand this virus and the next pandemic that will inevitably follow. In the short time that we've been paying attention to this infection, we've already made some important advances. There was a recent paper from 2 of our research groups, Alex Sete and Shane Crotty, and with other collaborators. And people were worried that if you have a mild infection, maybe you're even asymptomatic, that you wouldn't make an immune response, that the virus would have ways of evading immunity. But what their work shows, and this is just an example of one of the graphs from their recent paper, even a mild infection can raise strong immunity. So here's the control, and these dots up here, each dot is a person. Each person was making a measurable immune response to SARS-CoV-2. There is some T-cell immunity from other coronaviruses that cause colds. Not so much antibody immunity, but there is some immunity because of that. And it's possible that that immunity is beneficial. We don't know that. And it's still possible that in some severe cases, the immune response actually might be overreacting. But regardless, what this early work shows is that we have the tools to measure what's going on and that a vaccine should be feasible, that this virus doesn't completely evade the immune response. And this work has gotten us a lot of attention from major media and other internet outlets. So to make advances, we need to have the best scientific instruments. And some of our recent purchases are shown here for gene sequencing, for visualizing viruses and molecules, and instruments that allow us to analyze and detect rare cells. And this represents a capital investment of around $11 million. And you'll actually see some of these instruments later on. So we still wish to improve our infrastructure. We're making investments. Particular need is to fully outfit a biosafety level 3 facility, which will allow us to grow and analyze large amounts of the SARS-CoV-2 virus. We have the room set up. There's still some equipment that we need, and we want to expand our ability to collect blood from individuals, and while keeping those individuals safe and maintaining the appropriate social distance. So there's some other changes to the building that we require, and not shown here is a big ask. We do want to get a mass spectrometer, which will allow us to analyze proteins, and that's another rather expensive piece of capital equipment. So we're seeking funding for these items and these building changes, and of course, philanthropic gifts have allowed us to make the advances that I've very briefly summarized, and to turn our attention very rapidly to this important problem. Also, we're interested in having people enroll in clinical trials, and particularly individuals who may have recovered from the virus. So you can go to our website and you can learn more. As you can see here, you can scroll down you can say, I'm interested in participating in a COVID-19 study, and then you can give some information. What we're really interested in is those individuals who have recovered and been diagnosed with COVID-19. And of course, anonymity will be protected of the individuals. But we really want to know how people who've been exposed to the virus have responded, and what parts of the virus that they're responding to, and what kind of immune response they're making. Okay, so without further delay, I hope that puts a context on this for some of you. I know some of you have attended these before, and our speaker today will be Dr. Erica Ollmann Saphire from the Infectious Disease and Vaccine Research Center. And Dr. Saphire has brought the amazing cryo-electron microscopy technology to the Institute and enabled us to acquire the cryo-EM instruments. Dr. Saphire has been asked by the Gates Foundation to create a consortium to evaluate the therapeutic potential of antibodies, as she will tell you. And she's also been asked to act as part of another consortium that involves the NIH, major companies, and the European Medicines Agency that will also be involved in picking those antibody therapeutics that will be going into clinical trials this summer. So in short, Erica is a remarkable scientist and individual who will have a big impact on which antibody therapeutics will be going into patients in the future. So thank you for coming. Thank you for your attention. And I'll hand it over to Erica.
Thank you, Mitch. So the title of the seminar series is Live from the Laboratory. But as you can see, I'm not in my laboratory, I'm in my office. Because my research laboratory has 30 people in it that are hammer and tongs in this coronavirus problem, and that creates a lot of joyful noise out there. So I'm in my office where you can hear me, and I'm surrounded by my things. Now, you've heard people say that they wear a lot of hats. I say I wear a lot of jackets. You can see over here, I've got my white lab coat and my navy blazer. And over here, I have my academic professor robes. But the jacket that I like best is this one from the Coronavirus Task Force. It's a group of scientists and investigators here at La Jolla Institute that are understanding, harnessing, and learning what tools we need to build immunity against this novel coronavirus so we can all go back to work and school and human endeavor. So I'll show you now some of the new research that we have done in my group with an update. So here's where we are as a nation. We've now had 2.4 cumulative million cases and 125,000 deaths, with another rise on the way. Where we are in California is here with another rise as we go into the summer. And as we get some perspective on this schedule by putting in some dates. So for example, the shelter-in-place order was about here. It takes about 2 weeks for an infection to manifest as a case, because it takes about that long for the virus to incubate and cause disease. 2 weeks after shelter-in-place is right about here. So all of these cases are people that were likely infected right about the time we were told to shelter in place. Easter is here, and that's 2 weeks later. Here's Memorial Day, there's 2 weeks later, and another climb still. And we don't know what's going to happen, but we can predict with more movement and more human interaction, we're going to expect to see more cases. So the world very much needs vaccines to deliver broad protection and for us to feel safe going forward. The very earliest estimates are that a vaccine might be available in January. Most estimates are later, often July to September. Either way, we're looking at 6 months to a year or more, likely in that year range, for vaccines to be available. And the other important date is not the date that the first person can go into CVS and get a shot, but the date that we have broad vaccine coverage, because it's been very clear this year that virus anywhere rapidly becomes virus everywhere. We're not protected until we are broadly protected in all places on the globe. Even once the vaccines are first approved and first available, they're not going to be in all places at all times. We're not done until we're all protected. And even when vaccines have been made available, there's this significant issue. There are people who will have not been vaccinated or just not vaccinated yet. And there are those groups who can't be vaccinated. For example, pregnant women, the immunocompromised, all babies under age 1 cannot receive certain kinds of vaccines. And so their protection relies on you, relies on herd immunity. We can't have herd immunity without broad protection. Also, sometimes the vaccine just doesn't take, even if you've gotten it. So we need something else. We need something complementary. We need something very safe, and we need something immediate. And that's something I would propose would be antibody therapy. Now, antibodies are what your immune system makes after you have survived an infection. You're infected with the virus or vaccinated with the virus, but no one's been vaccinated with this virus yet. And then days to weeks to months after that exposure and your survival, your immune system makes and then improves antibody molecules. These are these little Y-shaped structures I'm showing you on the right. And the job of antibodies is to do 2 different things: to seek out and directly anchor onto a pathogen like the virus. Once it is anchored on, they can inactivate or neutralize that pathogen and recruit immune system help, recruit the cells of your immune system to come and clear that virus from your body and clear those infected cells so they stop being factories to make more virus. Antibodies are remarkably precise. They will hit one particular place and one particular pathogen and seek out just that one thing. They have 2 arms and a base. At the end of the arms are combinatorial loops. that are hypervariable in sequence. That's what evolves to give an antibody its precise specificity. So that's your molecule. These would represent the different pieces of different viruses. At the end of those arms are like fingers that have fingerprint-like recognition for a particular pathogen on a particular place. Once they anchor on, they could do something like block that virus from attaching to your receptors, or block that virus from being able to drive itself in, and then recruit immune system control. Antibodies are so powerful at their protection that you can deliver them as a drug. We're all familiar with the concept of antivenom. Antivenom is antibody against the toxins in snake toxin. You know, if you get antivenom right away, your life will be saved, your tissue will be saved. And with antivenom, you know, earlier is better. The same thing with antiviral antibody. Earlier infection is better, and what it can do is dramatically knock down the amount of virus you have, clear the virus, clear the infected cells to give your immune system a fighting chance to catch up and clear the rest. Now, I've shown you cartoons of Ys and puzzle pieces. This is what it really looks like. So in gray would be, for example, the spike protein on the surface of the virus. In yellow and red is that Y-shaped antibody, but that Y is coming out at you. So these are the two arms that end in those fingerprint-like recognition loops. Those are called FABs, fragment antigen binding. And this is the base going into the screen. That's what recruits your immune system. That FAB anchors onto a particular place on the surface of that spike. And there's a particular surface shape complementarity and chemistry that allows that one particular antibody to anchor to that one particular place with precise recognition. Antibody therapy has been used not just for snake bites, but also for other infectious diseases. We have antibodies against respiratory syncytial disease that are used to protect premature newborns from that seasonal winter respiratory virus. We have antibodies we have mobilized as treatments for autoimmune disease and for cancer. And it offers a remarkably precise mechanism of therapy where an antibody could seek out and destroy an individual cancer cell that you couldn't get at with a scalpel, or you might not be able to find any other way. And because antibody is a natural human immune molecule, it's what you would make yourself had you the opportunity or had that immune response inspired. It's very safe. It's also fast. IV transfusion of antibody will give you some degree of immunity immediately, just as fast as it gets into your veins. It's transient. Antibodies typically last about 3 weeks of a half-life. You can engineer them to last several months. They can be used to prevent someone from becoming infected or to treat people who have already become infected to prevent them from progressing to severe disease. But because it is a natural human immune molecule and it doesn't involve anything foreign, it is very, very safe. And it is often a de-risked therapy and one that you could give to nearly everybody. The challenge is finding the right antibodies to give as a drug. You have a quintillion possible antibodies in your sera, and they're against everything you've ever been exposed to. Among those in a survivor of COVID-19, maybe 1% are against that virus. And among that 1%, there are some that are extraordinarily potent, and there are other ones that are duds. You want to find the ones that are most potent. If you can find the very best 1 or 2, sometimes 3 different antibodies, you can characterize them very well. You can even optimize them. You can manufacture them in GLP, GMP clinical facilities, and that becomes your drug. The challenge is, how do you find the right ones to deliver? Let me go back. Right now, there are 50 to 70 different companies racing to find what they think are the best molecules that they can mobilize for antibody therapy against SARS coronavirus 2. Each one of these companies, whether they're a multinational corporation or a small biotech or even an academic lab, is going about it in a very slightly different way. Some of them are using different bait, different forms of the virus or different forms of the viral molecules. And with different bait, you catch different fish. So they're going to select different antibodies against different sites. They're using different strategies to evaluate them. Each one has set up their assays in different ways to look at different candidates. And some of them have chosen to optimize either those FAB arms and their fingerprints or that base that recruits immune system protection to try to steer the immune response. So every one of these candidates is a little bit different. And what we want to know is which ones are best. And that can be a challenge to know which ones are best because it can be an apples to oranges comparison. So let's say you are in charge of organizing a clinical trial and you have a limited number of clinical trial beds and a limited number of dollars to spend on that trial. You want to know which are the therapies that have the greatest likelihood of success, which are the ones that are safest, and which are the ones you can choose. You can't test absolutely everything because you don't have enough beds and money for that. And also you need enough patients given a therapy to get statistical power, enough people at different stages of disease or different ages and different genders. How do you choose? Well, if company A says we think we have the best therapy, and we know because we evaluated it using experiment A, then company B says we think we have the best therapy and we have evaluated it using experiment B, and company C says, well, we have a great therapy and we know it's good because we used experiment C. When you're comparing experiments A, B, and C, and even if it's the same kind of assay, if it's set up in different labs using slightly different conditions, the results may not be comparable. It's apples to oranges. What you need is that apples to apples comparison. You need to be able to compare all of the different molecules being raced forward by all of the different companies and academic labs to figure out which really is the best. Now here's the other thing. Viruses mutate. A virus will mutate to escape any single thing you throw at it. So often, what we use is a combination of things. We use maybe 2 different antibodies together that would hit different footprints. And by hitting different footprints, they make it harder for the virus to try to mutate 2 things at once. It mitigates escape. Also, if you're using antibodies in combination, you can find molecules that have a synergistic effect. One helps the other, or an additive effect. You can knock out the virus in 2 different ways. So we need to look for combinations. And the best combination might be 2 molecules raised in 2 different labs, and you never would have known about that possible combination unless you had a way of putting them together in the same place. The other issue is how to make this affordable. Antibody therapy is more expensive than vaccines. It can be more difficult to deliver broadly, but broad is what we need. It is very clear for these viruses, and especially respiratory viruses, that a few infections in one remote place have rapidly become millions of infections everywhere that humans live. We need to be able to broadly deliver this therapy, including to people who cannot afford it themselves. How do you achieve broad delivery? One way is to try to make the molecules more potent. If the molecule is more potent, you don't need so many milligrams. So if you can manufacture, it can spread across more people. It can also make the molecules more durable, more stable, so that they will grow to higher yield and you can just make more. By lowering the cost, you can also deliver it to more people. And there's different markets here. There is a market in the developed nations for the best therapy you can make no matter what the cost, but we also need to make sure that we are broadly protecting people that are in low and middle income countries as well. So how will we do all that? How will we compare all of the different antibody therapies side by side under the same assay conditions, find the best combinations, figure out how to optimize them, and make sure that therapies can be mobilized anywhere in the world and have that right safety profile and that right durability and the right manufacturability? We need to look from among the hundreds of therapeutic candidates being raced forth to figure out which ones are the very best. We need to analyze the very broad playing field, no matter where these antibodies are being made. If they're made by a small group or a multinational corporation, we need to put them side by side and figure out how they work. In order to encourage participation, when companies have invested millions and millions of dollars in these discoveries, we need to protect their intellectual property. Even if the scientists would want to contribute their molecules to someone else's study, the university or the company's lawyers might not let them. We need to find a way that protects their investment, but still makes that molecule available for science and medicine. And we need those therapies to last. We can expect the virus to mutate. We want that antibody to remain responsive, or we want to know what we can bring out if there is a mutation event. We need this antibody to last because we're expecting seasonal return of this virus. We need durable therapies. And we need an extraordinary level of safety. Most people infected with coronavirus will be all right. We want to make sure that we are introducing therapies that have little to no risk. We want to make sure we can use these therapies in those people that cannot receive vaccines. We want to use these therapies in immunocompromised, in babies, pregnant women. And we need to work quickly. This virus is expanding rapidly. We can't wait. We have to work on a much more accelerated timeline than we ever have before. How do we do all of these things? How do we find a way of galvanizing 50 to 70 competing groups into a single force very quickly to analyze things side by side? The answer to that is this consortium here that I was asked to build by the Therapeutics Accelerator of the Bill and Melinda Gates Foundation, called CoVIC, the Coronavirus Immunotherapy Consortium. We are now gathering those therapeutic candidates being advanced all over the world here at La Jolla Institute, and we are blinding them. We're giving them all code names. That makes the study very fair. Nobody knows whose antibodies are whose. We're just analyzing by code name to figure out what are the characteristics that make that best. Now the program officers know the codes, and the people who own the antibodies know where their own are in the study. So they know if they want to keep investing in that molecule or if they think there might be one better. We are going to, from the therapies being advanced in the world, find the ones that are the very best, find among them the best combinations, learn how we can engineer them for greater potency and to avoid any adverse effects, and try to make them available. So what will happen first, the samples will come here and they will be blinded or anonymized. That is to protect their intellectual property. Now the companies know which molecules are theirs in the study, and they get all of the data we generate back. So there's a benefit to them because they can use all of that data for their investigational new drug filing. By participating in the study, they can gain complementary independent verification and additional data for kinds of studies to which they may not have access. We're going to then aliquot and distribute identical box sets of the world's therapeutic candidates to expert virologists and research organizations that are going to evaluate them in different ways. All of that data will come back and will be cataloged in a database we call CoVIC-DB that is being run by Professor Bjorn Peters at LJI. This will represent the largest, deepest, broadest data set of antibodies against coronavirus yet assembled. This is going to be a tremendous body of information that we can use to evaluate not only these therapeutic candidates now, but what will work in the future, how we might fight variants of the coronavirus, which antibodies might reflect those we would like to see from vaccines, and how it could respond to new emergencies in the future. From the candidate therapeutics and the data we analyze, we will select therapeutic candidates we want to move forward, optimize them, and produce them. The functional analyses we are doing are these. We're going to analyze how well they anchor to the surface spike, with on rates and off rates and kinetics. We're going to look at their ability to bind different variants of spike that we see. We're going to sort them into what footprints they have. It's called epitoping. We're doing high resolution structural biology to understand the chemistry of those interaction sites and what they look like, what context they rely on. We're understanding what each antibody does with that base of the Y, how it recruits immune system health. We're looking at the ability of each of these antibodies to neutralize the virus in different cell types and under different conditions. We're looking at the likelihood of each of these viruses to be escaped, where that escape mutant might be, and which ones might be less likely to be escaped and last a while. Their ability to confer protection in living things, combinations, and optimization. Our first goal is this. We need therapies in people. It's very much a product-driven goal. We want one or more highly potent combinations of at least 2 fully human neutralizing antibodies against this virus to prevent severe disease. We're going to optimize them to mitigate escape and deliver them broadly. But with my other jacket, that academic professor jacket, we want to do some discovery too. While we've gone to the trouble of finding ways to get these 50 to 70 competing entities to agree and contribute them into this very large study, we have an opportunity to really learn something. We can learn the range of different ways in which an antibody could try to defeat this virus. What's successful, what's not successful. Which antibodies are synergistic with each other? What determines how these antibodies are elicited? Do we see the same kinds of antibodies in vaccines or not? And we can use these antibodies for basic research discovery tools and understand how we might use this information to craft better vaccines. We're doing this in order to save lives. Our first use case is treatment. To find individuals with early disease, mild to moderate COVID-19, to treat them, to prevent them from progressing to severe disease. And so there may be people that we see early in disease that have the markers of the likelihood for progressing to severe disease. We can use these antibodies to lower the amount of virus they have and give their immune system a good chance to clear it, prevent severe disease progression. The second use is prophylaxis, preventing disease. You can give antibody to frontline medical workers, for example, or high-risk groups like pregnant women to prevent them from becoming infected. This is key for people who can't be vaccinated or people where the vaccine isn't available yet. Right now, that's all of us. You can also use antibody in a disease outbreak situation. So if the virus has emerged in a nursing home or in a village, you could treat the contacts and the contacts of the contacts to prevent spread from that epicenter. And it's starting to work. So this month here, the antibodies are starting to roll in. And this is my program manager, Dr. Sharon Schindel, now receiving, aliquoting, and standardizing these antibodies for shipment. So the goal of this consortium is to find one or more therapeutics so we can mobilize broadly to anyone in the world. But while we are doing this search, the virus has undergone tremendous human-to-human transmission with well over 9 million cases, nearly 10 million so far. This is data from the Johns Hopkins Coronavirus Resource. That yellow graph of cumulative cases in the bottom right isn't slowing down. As the virus circulates from one human to another around the globe, it has an opportunity to mutate. Every time a virus replicates, it's a roll of the dice whether it will pick up an error. Some viruses like Ebola or HIV have no proofreading machinery and they're quite error prone. Coronavirus, thankfully, has some proofreading capacity, so it doesn't mutate as quickly as those, but it does mutate. And with every replication cycle in every one of these people and every transmission, it's another roll of the dice. And so it's inevitable that mutations will arise. And they have. This is now data from Los Alamos National Lab. They deposited in bioRxiv, looking at mutations that emerged around the world. And one thing that caught their interest is a mutation in that spike protein to which we're targeting our antibodies. The original type of the virus had an aspartic acid, we use code letter D, position 614 in spike. This is what emerged from Wuhan. In February, a mutation arose, putting now glycine, G, at position 614. And within a very short period of time, the orange virus was replaced or superseded by the blue virus in the world. You can see there would become a much more abundant blue type of the virus, the G614 virus in Europe, North America. England had only the blue virus. And then if you look down in Taiwan, there's a second wave where they were infected with and then cleared the orange virus, but then the blue virus is reintroduced and caused more cases. So this virus emerged and rapidly spread in the course of a month, that one mutation. And so that's something we really want to keep an eye on, that mutation and others. The eye that we keep on it here at La Jolla Institute is this. This is our new Titan Krios electron microscope. It is not the kind of microscope you used in biology class to dissect a frog. It is 11 feet tall. It is the most powerful microscope commercially available. It shoots an electron beam of 300,000 electrons down the center of the column. You're watching it being constructed here. The power of that electron beam and the precision of the detectors that can pick out each individual one of those electrons raining down on the sample allow you to, with unprecedented resolution, see something as small as individual interatomic distances. We can, with extraordinary precision and resolution, understand the precise fold and structure of the virus and the molecular surfaces by which the immune system defeats it. We can see where the rubber hits the road, not just for antiviral immunity, but everything that we want to study for human immunology, now at beautiful and clear resolution. And so this is a map from this microscope, like ours, of the surface spike of the coronavirus. This is the molecule that the antibodies attack. This is what creates the corona on the surface of the coronavirus. You're looking at a topographical map of where its electrons are produced by cryo-EM. If we dive inside, we can build the model of how the genetic information in the RNA encodes a polypeptide and the structure of each amino acid side chain of that polypeptide. In red is that aspartic acid at position 614. By substituting that with the glycine, we lose a hydrogen bond to the neighboring molecule. We see that that change occurred. We see that that change dominated throughout the world. But what does it mean? What happens when the virus does that? Well, to figure out what happens when the virus does that, we made pseudoviruses carrying the old spike. We looked at their ability to infect different kinds of cells in the lab. So monkey cell line, a human cell line, all sort of infected with a protease, that's a clipping enzyme that the virus needs. We could see the level that they could infect in these cells. When the virus has achieved that one tiny mutation that became dominant throughout the world, it infects 2 to 3 times better. It has a much higher titer. That new virus, the blue virus, is fitter. It's more robust. That's what happens in the lab. What happens in people? Well, in people, you measure the amount of virus by something called the CT value. That's the cycle threshold. You measure the amount of virus by seeing how many cycles of PCR amplification are needed to detect the viral RNA. Only a couple cycles, there's a lot of virus. Many cycles, not so much virus. We look at patient data from Sheffield, UK, when the virus emerged. You can see that the blue virus has a lower CT value than the orange virus. The new virus grows more robustly in patients as well as in test tubes. There are fewer cycles of application needed to detect it. So are we in trouble? The virus mutated in March. That mutant virus spread throughout the world. It grows 2 to 3 times more robustly in the laboratory. It grows more robustly in humans as well. Well, to see if we're in trouble, we looked at the ability of human antibody to inactivate it. So we looked at San Diegans who recovered from coronavirus, and they were all infected early to mid-March when the D virus and the G virus co-circulated. You can do a neutralization assay. How well can all the different antibodies in their blood inactivate the virus? Inactivate the orange virus, inactivate the blue virus. So to read a neutralization assay, you look at it this way, where on the y-axis, on the left, is the relative infection of cells, from no infection at the bottom to complete infection at the top. And on the bottom, the x-axis is the dilution of antibody. So on the left, that's your antibody from that blood straight up. And on the far right, that's highly dilute. So diluted 10-fold, 100-fold, 1,000-fold, 10,000-fold. To read these curves, you might look at the amount of antibody needed to neutralize half the virus or neutralize 90% of the virus. And you look at the points required to do that. At each one of these points, you can see the blue curve is pretty much in the same place or slightly to the right of the orange curve. You need the same or actually a little less antibody to neutralize the new virus than the old virus. But that's one person. What happens over several? Well, if you look at all 6 people, you get the same thing, where every single one of them, the blue curve is roughly in the same place or slightly to the right of the orange curve. Even though the virus grows 2 to 3 times as robustly in cell culture, you don't need 2 to 3 times as much antibody to neutralize it. So that's really good news. That new virus that took over the world is not harder to neutralize. Human antibody, your human immune system, is up to the job. Now that is, of course, these 6 people, and we're going to need more studies to see if this is true for all people. And we need to see more studies to know if the vaccines being developed, which were all developed by that, using that orange D reference strain, elicit responses that remain applicable. The other good news is not just that the human antibodies can neutralize the virus in the test tube, but it doesn't cause a worse patient outcome. If we looked at patients in Sheffield that are infected with the old virus or the new blue virus, we compared who could be treated as an outpatient versus an inpatient versus in the ICU, there's no real difference in patient outcome. So it's a fitter, stronger virus, but it doesn't seem to cause more disease. It does, however, grow more copies of itself, creates a greater magnitude of infection. And so the worry is, maybe, even though we dodged a bullet there, could it propagate another mutation that would make it more lethal or even more transmissible? What was alarming about this finding is that a mutation here at this time point became globally dominant a month later. That's not a lot of time to respond to things and change things. This is why we need a large database of an arsenal of antibodies so that if a mutation emerges, we know we have another one to replace it to. With this much human-to-human transmission and that many rolls of the dice, the virus will mutate again. There is and will continue to be human-human transmission for a very long time. What we need is immune defense. To get out of our houses, to go back to education and jobs and social interaction, we are relying on immune defense. Whether that immune defense is your own human immune system or what we can do to inspire it to be better, the vaccines or antibody therapies or treatments. We need to discover these rapidly. We need to de-risk them to make them very safe, and we need to evaluate them and find out which are the ones to mobilize. The study of human immunology is more critical than ever and has been focused in three major areas. We might be researchers in infectious disease and vaccines. We might be researchers in cancer. We might be researchers in understanding autoimmunity or inflammation or allergy. But the important thing to know is the interconnection of each of these areas and how all of them can influence and learn from each other. So antibody therapies against a human molecule called IL-6, discovered in very basic analysis of what constitutes an inflammatory response, can now be used to dial down that severe acute respiratory disease and severe COVID-19. So a discovery from basic inflammatory biology can be used to treat that infectious disease. Antibody therapies evaluated for cancer that target integrins might also be used to treat COVID-19. And in the same sense, by studying these infectious diseases, we understand how they trigger other things later on. There are a number of viruses and microbes known to trigger autoimmunity and inflammatory disease. 24% of known cancers, and I'm sure there are others that we don't know yet, are caused by a viral infection. For example, there's a well-known link between human papillomavirus and cervical cancer. Epstein-Barr causes Burkitt's lymphoma and stomach cancer. Hepatitis B, hepatitis C, two entirely different viruses, both cause liver cancer. Viral infection has also been known to inspire different kinds of autoimmune disease. Some of these we have links for. Other times there isn't a smoking gun because that viral infection was 10 years ago. But here's the important thing. By studying these viruses, we can develop a vaccine. If you have a vaccine, you can prevent that later cancer. That's the kind of advance we need to make. The other thing is that the study of viruses teaches us how our cells work, and they teaches us how our immune system works. That virus is not alive. It needs your cell to propagate. In order for that virus to propagate itself, it must use and hijack and silence the genes of your cells. Those viruses know our cells better than we do. They know them better than science does. By following what a virus has chosen to silence so it can replicate unchecked, or what a virus has chosen to amplify to increase coughing and spread, we can learn more things we need to know about our basic immunology. We can understand better how our cells work. We can understand how viruses control inflammation, so we can learn how to better control inflammation. So an investment in any one of these areas improves all of them. And that is the wonderful thing about the think tank where we work, that we have these 21 investigators that each have a different focus in the lab, but the basic knowledge they make or the translational discoveries that the research advances can amplify every one of these other areas. Because even though we have a different focus, it is one human immune system with a power and complexity that we're only beginning to understand. And the power and the complexity of that human immune system is what can get humanity out of our houses and to be done with this virus. So for support of this research, I'd very much like to thank the Therapeutics Accelerator of the Bill and Melinda Gates Foundation, MasterCard, Wellcome Trust, and others for supporting that broad CoVIC study. I'd like to thank the Overton family that supported all of that research on the mutant. When we saw that mutant emerge and how quickly it was spreading, it was very urgent that we understand its effects right away. And there really wasn't time to go and write a grant and try to get it funded. We had to act immediately, and they made that possible. I'd like to thank the individual people in my lab, Sharon Schindel, the program manager of the CoVIC, which has been a tremendously sized organization to grapple with and coordinate and move forward. Michelle Moss, my laboratory program manager, Catherine Hasty, an instructor who did the neutralization studies I've showed you on the mutant, Alex Moonwalker, our graduate student, for also analyzing the effects of the mutant, Vamsi Rayaprolu and Ruben Diaz for generating the graphics and the EM and powering the structural biology behind that CoVIC program. And I'd like to thank you for your attention. And Mitch and I are happy to answer any questions. And I'm excited to go down and see behind the scenes as well.
Thank you. Thank you very, okay. Are we, we're going to questions first, then to the microscope? Yeah, I guess so. Okay, first question. Microscope first?
Actually, we're down here and ready. So if you can hear me and see me, all right.
Let's do that.
So, thanks. Thank you, Dr. Saphire. I'm here just off the atrium inside the Institute, where a year ago we were talking over blueprints about a cryo-EM facility and an advancement in our BSL-3. And because of the partnership and the funding from philanthropic donors combined with federal support, today I'm actually sitting in the control room. So think of it in the same way. If you were to look up into the night sky with a giant telescope, there'd be a control room. Here we're looking into the inner world where the action really happens. And I'm not going to mess around with the controls, but in brief, what this is we're looking actually inside the viewing chamber of a transmission electron microscope. And what might strike you is the similarity to astronomy, where you see what looks like stars and shiny pieces. In fact, we're looking down at the nanometer scale. So just tiny fragments at a point where if we zoom in, we can actually get into those areas where in a very physical way, things touch, things work. This is the information we need. Now, this control surface and the highly technical staff that work on it end up creating fields of information like this out of thin slices. But where the additional magic happens is in this next figure. This is what we see out in science, where the entire globe, the entire field of research can take a look at these representations that have been derived from the machine right out there. And now the entire scientific enterprise can take their shot at it as well. And so I'm going to pass now over to Dr. Diaz, and he's going to give you a look inside the machine itself.
Okay, my name is Ruben Diaz-Avalos, and I'm the Director of Electron Microscopy here at the La Jolla Institute. Here we have a state-of-the-art cryo-electron microscope. As you can see from, this looks like a box at first glance, but then looking inside, you can see kind of all the guts of the microscope and everything. And one thing that you can see is that at the top is where the electrons are generated. So that is all the way there. That's where the electrons are extracted, and then they are accelerated to 300 kilovolts so that they hit the specimen that is in this area. And then everything else is going to be lenses essentially that are forming the image, which then pass through a prism that sends them to the back of the microscope where they are detected into an image. So in that way, we can generate images with a very exquisite detail, and we can see details up to atoms in ideal cases. The reason we use electrons for this kind of imaging is because the resolution in any microscope depends on the wavelength of the illumination that you are using. And certainly you can use light that has the correct wavelength, but the problem is that for X-rays or that kind of illumination, there are no lenses. However, electrons have a 300 kilovolts. They have a wavelength that is the order of picometers. And then in that way, we can probe very small distances, and then we can get images with atomic resolution in ideal cases. Now, the way that we do that is by putting the sample in what we call an electron microscopy reel, which is a very tiny wafer, essentially made of copper. And then this wafer is frozen immediately with the protein. And then once the protein is frozen, we put it in a gadget like this that is essentially a door that keeps the specimen cold at the liquid nitrogen temperatures. Then with the specimen being this cold, we simply transfer it to the electron microscope. And then there is a robotic arm that picks up the specimen and then keeps it inside the microscope at liquid nitrogen temperatures. Now, also you might wonder why is it that we need to cool down the specimens? The reason is that the specimens, when they are illuminated to get an image, they get a lot of radiation, and radiation destroys them. The amount of radiation that they get for one image is the same as if they were about 20 meters away from a thermonuclear blast. So you can imagine that that's going to essentially vaporize them. And in many cases, you don't even have time to get a picture. By cooling them down to liquid nitrogen temperatures, we have a resistance to radiation. So it's kind of like putting sunscreen on your proteins. And then in that way, we can actually build an image that can be processed afterwards. So one thing is that we imagine if you are having a bad day, imagine the kind of day that the specimens are having. So I guess we can go back now to Dr. Wilson.
Thank you, Ruben. So unlike the cryo-EM facility and the control room that was right off the atrium, I'm now back down a long corridor that's off the main walkways because we're going to be heading into the biosafety level 3 high containment lab. And before we go in, I'll just note that we're able to go in today because we're in the middle of recertification. Normally, I couldn't go in like this and do the work. And so I'm just going to give everybody here kind of an inside look as to what it's like when you're going into a facility that's built to contain aerosolized, potentially deadly microorganisms, viruses, things like that. So only secure personnel are able to come in. As you enter into the BSL-3, there are lights and locks around me that won't let me go into the next part until that one clicks closed. There are pressure sensors to make sure that the airflow is always pulling clean air into the facility and it's exiting up and out. So there's no recirculation. It's a one-way trip for the air and anything that might be in it to get through. We're next going in to the main area. It's clean and shiny, and it's going to be this way for a long time, as a matter of fact. It gets scoured and sanitized often. And as researchers come in, each of the individual rooms get assigned to various purposes and different pathogens that may be used. Everyone that comes into the facility, for instance, gets a full understanding of what's being done in here so that they can think about and protect themselves at every moment and think about things as they leave. If you turn the camera that way, everything except for the people working in here, once it comes in, only leaves through an autoclave. It gets cooked down and sterilized. So the entire build of this room actually requires that it is an institute in itself, that it has all the instrumentation, all of the equipment, everything that it needs inside these walls. It's building that mini facility. And this is why we've asked for so much help and gotten it at times for new instruments specifically built for these very, very dangerous pathogens. There are standard pieces of safety equipment you'll see in labs throughout the Institute. And then there are some things that are just very purpose-built that only in a BSL-3 you really need. One of them is major alarm systems for leaks, security cameras to make sure that there's nothing happening in here that we can't monitor. And finally, as we head out this way, the exit system. This is where, after time in the DSL, staff will come in and actually take off the clothes and protective equipment that they've been wearing.
Oh. Hello. I think we've lost Steve. Okay. Well, let's, Erica.
Area.
Oh, Steve, we lost you for a bit.
Oh, I'm
So
sorry.
why don't you wrap up quickly and we'll go to questions.
Okay, you got it. I'm on my way out, and so we are.
Well, we hope you're not on your way out. Never mind. Okay. Erica, can you unmute and let's address some of the questions from the audience. Okay, so one question that often comes up is if you test positive for antibody to COVID-19, are you protected?
Maybe, but we are not sure, and we don't know for how long. So what I mean from that is you are more likely to be protected as somebody that has no antibody at all. But we do not yet know exactly what titer of antibody or how much antibody is needed to be protected. It looks like from the convalescent sera studies that if you can neutralize at about a 1 to 150 dilution, you might be good. Now, the second issue is that while antibodies against some diseases last your whole life, like polio and measles, antibodies against coronaviruses sadly do not. Now, this is tremendously individual. For many people in this current outbreak, their antibody has diminished. Now, this is looking at the original people infected earlier in the outbreak, so likely China. About 10% never mounted much of an antibody response at all. About 70% lost it within about 2 to 3 months. That tended to correlate with people that had a more mild infection, typically were more likely to lose their antibody. That is maybe different from T cells. But I think that's important information there for a vaccine, because we need to find vaccines that will stimulate the right artificial danger responses so that you can get that long-lasting memory response. The other thing we don't know is we measure circulating antibody, which is different from memory. In order to be protected with reinfection, you can rely on that memory reamplification, which could happen even if less seems to circulate.
So if antibodies diminish, why should we be hopeful for vaccine? And I think your answer already touched on that, but
yeah.
Maybe you could amplify that.
So a vaccine elicits more than antibody. A vaccine also elicits a T cell response. A vaccine can be precisely designed to do better than a viral infection. You can design a vaccine to give you a better, stronger immune response than you might get from that infection. Of course, it's a lot safer to get a vaccine than to get one of these lethal viruses by themselves. For some viruses, we need booster shots. And for some viruses with a lot of drift and sequence change, like flu, we still need seasonal shots. So a vaccine could still help. Worst case scenario, you just get your seasonal coronavirus vaccine with your flu vaccine.
Yeah. So I think we'll take 2 more because, I've just got, okay, we can go until 1:10 Pacific. So we can take a few more. So here's, you're doing great. By the way, fantastic talk, Erica, and good job to Steve and Ruben as well. So here's one. How will the results from the consortium from CoVIC be translated into clinical trials? And do you have any idea on when that will be? The when question often comes up, of course.
Yeah, it's immediate. And so the whole goal of this is to provide the broadly useful information that everybody needs. And the point of Bjorn Peter's database is that there's publicly accessible immediate information where as soon as we get it, it's available to scientists and clinicians and all stakeholders. The program officers that need to make clinical trial decisions have access to the unblinded samples. So there is a way that we have specifically designed to make sure that the information we see on that can immediately be used.
So how do you test for a neutralizing antibody? And do you use this? Well, I won't get technical with pseudoviruses, but which tests are better, or do we know which tests are better?
Well, that's a great question. So we ran a similar consortium for Ebola virus to answer that. And what we did is we decided that we as a field didn't really know. And so we ran every antibody through each of 34 different tests and figured out at the end, not only what were the best antibodies, but what were the best tests so we could do it faster next time. And so we were looking at the ability of each one of these antibodies to neutralize authentic virus in laboratory workhorse cells and also relevant cells like lung epithelial cells. We're looking also at their ability to neutralize what we call pseudoviruses, which are safe laboratory viruses that anybody can use, to see if those assays correlate with neutralization of actual virus. So far they look like they do, but we do see differences in how you set it up. So the antibody might help in one cell type and not in another, because viruses have different strategies of getting in. So we're doing lots of complementary assays to get that more complete information.
Are you, a little bit off the topic of antibodies certainly, but are you optimistic about some of the treatments that are now being tested for people who are ill and might be in the intensive care or even on a ventilator? Because those are also immunomodulatory, well, that's a lot of syllables, treatments.
Good Scrabble word. I am optimistic about those. You know, you need a different kind of therapy early in infection than later in infection, because early in infection, what you need to do is knock out the virus. Later in infection, you need to control an overactive inflammatory response that's what's causing that acute respiratory disease. And so there's antibody therapies against immune modulatory mechanisms, and those are going on in clinical trials, I think, in 4 countries. I'm pretty optimistic about those. And then also we had some good results about one of the steroids this week too. Yeah,
that's one of the surprising things about this, especially to people who aren't used to thinking about the immune system, is that a lot of the very ill people have a kind of distorted and overactive immune response. Distorted in the sense that the immune response has many arms, and one arm is doing too much work, if you will, and that's an issue. Okay.
We complementary studies from people looking at different arms, people looking at antibodies, people looking at T cells,
sure,
looking at immune memory and innate immune danger signals.
Okay, so a friend of the Institute, I won't mention any names, I got this on my phone, wants to know why there is such a difference between the spread in outdoor venues and in indoor venues, and maybe we can discuss a little bit about aerosols and droplets. And fomites, if we wish.
Fomites. So this virus, there are different kinds of viruses. There are viruses that have a hard protein shell, and they're very hardy and they're very sturdy and they're nearly invincible and they can last a long time. So norovirus, that cruise ship diarrheal virus, is difficult to kill. Adenovirus, a hard protein coated common cold virus, can blow from one village to another. Now coronavirus is different. It's the type of viruses that have a soft membrane outer layer. It needs to be moist and it's delicate, and you can kill it with soap, UV light, and heat, enough heat. So in order for that virus to transmit, it needs to be in a moist droplet. So the little respiratory droplets. And when you speak loudly or sing or cough or sneeze, you project these several feet. And so outdoors tends to be better. There's often more space to distance ourselves so we don't get within the 3 to 6 foot firing range of respiratory droplets. And also because there's UV light, there are breezes and air circulates. Where we've seen a lot of human-to-human transmission are when humans are confined together in areas with poor circulation or that circulate air from one apartment to another in a high-rise building.
Yeah, I avoid choirs. Unless you can sing through a mask, I guess that could work. So if I could summarize, and correct me if I'm wrong, antibody therapies can be faster and they can protect immediately, but they're going to be for specialized people. So the very young, the very old, the frontline health workers, et cetera. And the vaccines can, if we develop one, can be given broadly, they're relatively, in most cases, relatively inexpensive to manufacture compared to antibodies, and can provide their advantage would be long-term protection and the ability to literally give hundreds of millions of doses at some point. Is that a fair summary, or would you disagree with that?
Close. So vaccines are typically cheaper and can be given more broadly. Antibodies are great for people that haven't been vaccinated, or importantly, can't be vaccinated and haven't yet been vaccinated. So the goal of antibody now, even once vaccines are available, there have been people that haven't gotten them yet. Right now, none of us have had it yet. So you don't have to fall into a special category to qualify yourself for an antibody therapy now. If there's enough available, the goal is to just treat people early in infection. If supply is limited, they might need to steer it toward the people that need it more, people that are older or immunocompromised or have a likelihood of progressing severe disease.
Okay, well, my producer told me to ask the last question. And it is, what else do you need to carry out your work effectively? And before you answer, one thing we need is a mass spectrometer, but in order to diagnose fragments of proteins and how different cellular proteins are interacting with COVID virus proteins and so on. So I shouldn't ask a question and answer it. So I'll shut up and I'll let you give an answer.
We need that mass spec and we need that BSL-3. And the reason we need that mass spec is to give us precise single molecule analysis of what individual molecules, what individual things are turned on and off by this virus, so we can get broader resolution. That mass spec can also tell me which of the different varieties of carbohydrates stuck in that spike protein affect how those antibodies work. So viruses cover themselves with human sugars in order to be a wolf in sheep's clothing, but they put different sugars in different places. You don't get that information by sequencing it, also by solving the structure. We need mass spec to see what variability in those carbohydrates affects the utility of our antibody therapy and affects the kind of immune response elicited by a vaccine. We've got to have that mass spec. We also need that biosafety level 3 facility. We have a great new mouse model here in Susan Shresta's lab that could better evaluate whether the antibodies used for therapy or elicited by a vaccine could cause unintended enhancement of disease. But that has to be held in that specialized facility that Steve showed us. So we just need a few more pieces of equipment down there to get that running and be able to do those studies.
All right, Erica, thank you very much for that wonderful talk, and thank you for demonstrating the microscope, Steve and Ruben. We'll have this has been recorded, and we will be able to send it out. And there'll be a last slide not from me, I believe, correct? There'll be a last slide, yes, that will give people more information about how they can help and how they can contact us. And there it is, www.lji.org/COVID-19 for information on research and for information on help, donate, and also you can contact Chris Lee, our Chief Advancement Officer. So thank you to the audience for your interest, your very sharp questions, some of which I tried to answer during the presentation. Thank you to Steve Wilson and Ruben Diaz-Avalos, and thank you most of all to Erica for a wonderful talk. And everybody, stay well, stay healthy, social distance, wear a mask. Unfortunately, this is not going away soon, but take care. Bye-bye.